Okay, welcome. We are now beginning Unit 13. You're starting with Part 1, and in this unit we're learning about a new nucleic acid. This time it is the RNA molecule. So before we begin, and this is not a slide that you need to write in your notes, just a quick review of what we know already. From after, we know that DNA is the genetic material. It's the transforming factor, it's what's found inside the cell, it's what carries all of the information that gets passed from generation to generation. We know that this code, or this secret message, is carried in the bases, and it's how those four bases, the A, G, C, and T, line up that determines when information is going to be passed on. But what we don't know is exactly what does this code mean. Only four letters, and they determine every characteristic of every living thing everywhere. Before we talk any more about RNA, I wanted to take a moment to talk about two scientists, George Beadle and Edward Tatum. And even though they did not make it into your textbook, George Beadle, right here, born in Wahoo, Nebraska, of all places, and went to UNL. And these two men did win the Nobel Prize for what was called the one gene, one enzyme hypothesis. And what their idea entailed, basically, was saying, okay, we know that DNA has all of the information and has all of the instructions. Beadle and Tatum's work said that on that piece of a chromosome where all of this DNA was, all of these little chunks were responsible for coding or having the instructions for one particular type of protein. And that was a big deal because proteins, and we are going to see that in this chapter, are the key. They are what is responsible for determining every characteristic about you is determined by a protein, which a protein is made by a string of amino acids, which are going to come from instructions on the DNA, which it gives to the RNA, as we're going to see. So our big question now is, okay, DNA has the information. We know that. How do the rest of the cells get this information? How do organisms know what they're supposed to look like and how they're supposed to grow, reproduce, and act? Well, what we need is another nucleic acid. Remember, DNA was deoxyribonucleic acid. Our new nucleic acid is RNA. And we need it because DNA inside the chromosomes in these eukaryotic cells is in the nucleus, and it's unable to leave. So it cannot give that information out. It can't transport that information. And it's kind of like creating, your book uses the analogy of a master plan. So if you created this plan to build a house or build a city or whatever it is that you're going to build, or maybe you're simply just writing this wonderful novel and you had your original copy and that was it, you certainly wouldn't send it out and let anybody else just have it. Instead, you make copies and you pass them out. And those copies that you really don't care about are the RNA. And the RNA is able to leave the nucleus, travel throughout the cell, and convey the message from the DNA to the other parts of the cell. And ultimately, RNA's job is to make the proteins, which I've already said are so important because they are going to determine every single characteristic that an organism has. So you could think of the RNA as like a blueprint. The architect draws up this master plan. It's beautiful, it's wonderful, and then she makes all of these copies that she can present to all of the different workers who need to do all of the different jobs to make this building. Or maybe you've made copies of your novel and you're going to have all of these editors look at it, but you're going to keep your original copy. So what is it that makes RNA so different from DNA? And it all begins with the letter. DNA, the D stood for the deoxyribose sugar. Well, the first difference between RNA and DNA is the sugar. So the sugar is no longer deoxyribose, it is just ribose, starting with an R. The second difference you can see from this image, DNA, over here, double-stranded. We know that already. But if you take a look at RA, it is only one strand. So the difference, number of strands. And the third and final has to do with the bases. When you look at the bases, you can see that both of them have C's, both have G's, both have A's. But here's the difference. In DNA, our thymine is replaced in RNA with a different base called uracil. So T's, wherever you would have a T, you are now going to have a U. So now we're going to look at the three flavors of RNA, or the three different types. And these are the three main types. There's actually another type, and we're not even going to worry about it. But the first type is called messenger RNA, and write that as little m, big R, big N, big A, mRNA. And its job, it's easy to guess, is it's like a messenger. It's going to take the information from inside the nucleus that DNA gives it, leave the nucleus, and carry that information to wherever it needs to go so that we can assemble these amino acids. The second type of RNA is called ribosomal RNA, or rRNA. And again, just from the name, you can kind of tell what it is. Ribosomal RNA makes ribosomes. And ribosomes are made out of two parts. They're made out of protein, and they're made out of ribosomal RNA. Pretty easy to remember. And our third type of RNA is what we call transRNA, or tRNA. And this is what it's going to take the information that says, okay, here's the amino acid I need. It's going to go and pick up that amino acid, and you can see on this picture, it's picked up the amino acid, and it's actually going to transfer it or carry that amino acid to the ribosome. And if you remember from way back early in the first semester when we were learning about the cell, ribosomes are where proteins are made. So that is the job of transfer RNA. When DNA made a copy, we called that process replication. Well, when DNA gives its information to RNA, we call that transcription. It's transcribing. And if you transcribe, you're writing down. So what's happening in this process is within the cell, within the chromosome, little chunks of DNA are acting as templates or little pieces of instructions, and it's going to take those little pieces of DNA, write their information on a complementary piece of RNA. So we're not making a second DNA strand, instead we're going to make an RNA strand. This next part should not be a surprise. In prokaryotes, this occurs in the cytoplasm. Of course it does, because that's where the DNA is held. And you can probably guess where it occurs in eukaryotes. Yes, it takes place completely inside the nucleus. Just as we needed an enzyme to allow DNA to replicate, in order to transcribe DNA, we also need an enzyme. But our enzyme this time is a new one, but it's RNA polymerase. So RNA polymerase is going to do many of the same things that DNA polymerase does. It's going to go in, it's going to separate the DNA, so we're not using DNA heliocase, we're using RNA polymerase. And then when the DNA is separated, only one strand of DNA will be used as a template. So we're making only one piece of RNA out of our double-stranded DNA. Now this DNA molecule is humongous. There's hundreds of thousands and millions of bases. So where would you start? Where would you start making your copy? And it's not by chance. Actually, a method to it. The place where you begin is identified on that DNA molecule, and there's a special set of bases called promoters. And the promoters send out a signal to the RNA polymerase, kind of like waving your arm saying, "Here I am, pick me, pick me." So the DNA strand is starting right here. The promoter, special sequence of DNA. So it's not an enzyme, it's just a sequence of DNA. And when the RNA polymerase recognizes that sequence, it says, okay, here's where I'm going to start transcribing. And from this point on, in this direction, we will start making an RNA strand. This DNA strand will come apart. Now there is a whole lot of information on a DNA molecule, even when we're just looking at little tiny slices. So just like when you write a paper, as you go back and you proofread it later, you realize that not everything you wrote was that important. There's things that you can take out and throw away. And that same thing happens with our DNA molecule. So here, at this point, we're starting with our DNA. The RNA polymerase has come along, it's separated the DNA molecule, and what we're going to do is we are going to make a pre-messenger RNA molecule. You can think of this pre-RNA molecule as kind of your rough draft. So we had our good DNA, made a copy. Not everything in that copy was super important. We did not need it for the particular protein that we make. So what happens in the cell is some editing takes place, some chopping. And it turns out that there are names to these parts of the messenger RNA. Names for the good parts and names for the bad parts. The parts that get cut out are called introns. And the way that I think it's easy to remember that is these introns stay in the nucleus. They do not get to leave. The important pieces, and you can see as we went, so we had our DNA step 1, our pre-messenger RNA step 2. These introns were snipped out, removed, and then the rest of the messenger RNA hooks back together, and the pieces that we do keep are called exons. And you can remember that because they get to exit the cell. The exons are what are allowed to leave. And then a cap and a tail is placed on that messenger RNA molecule, and it's good to go. It's ready to leave the nucleus. And as it leaves, we call it messenger RNA. So to put it all together and kind of do a little bit of what happened last chapter and look at what's going on in this chapter, and also to help you do your homework assignment, which is going to be worksheet 13.1, let's look at this DNA strand right here. So this is our original strand. And I know you guys are really good at this. What would the complementary DNA strand look like that goes with that? Hopefully I wrote it right, but you guys can check. So everything that was an A becomes a T, G's become C's, T's become A's, C's become G's. So our complementary strand looked like this. Well, the new part, and the only part that's different in this particular section, is what in the world is our mRNA going to look like? Well, it is going to look exactly like your complementary DNA strand, except for one letter. Do you remember what that letter is? It's the U. So anywhere there was a T on our complementary strand, we should now U in our messenger RNA strand. What if we don't know the complementary DNA? Then we're simply going to transcribe from the original DNA. So A's would normally be a T, but it gets to be a U. G's still become C's, and so on and so on. So I've put 2 other examples out for you to take a look at. On this one, I gave you the complement, and see if you can figure out what the mRNA strand should be and what would the original strand have been. And in the last section, I gave you the messenger RNA, and see if you can work backwards and turn it into complementary DNA and original DNA. And then if you can do that, you are ready to do the homework for this first unit. The worksheet 13.1 has some defining that you need to do, identifying DNA and RNA molecules in the different parts, and then doing some of this transcription. So go ahead and get started on that.